All right, indeed, let's get started. So hello, everyone, and a very warm welcome to our very first seminar of the 2022 Distinguished Seminar Series. And I'm delighted to welcome Professor Jinjun Shi from Harvard Medical School and the Center for Nanomedicine at Brigham and Women's Hospital. So Professor Shi's interests include nanotechnology, biomaterials, RNA therapy, and immunotherapy for transformative biomedical applications. His laboratory research is currently focusing on RNAi delivery for gene silencing, mRNA delivery for protein restoration, immunonanotherapy, and development of stimuli-responsive biomaterials. So throughout his career, Jinjun has won various very impressive distinguished awards, including the Prostate Cancer Foundation Young Investigator Award in 2014, the American Lung Association Lung Cancer Discovery Award in 2019. Jinjun was elected Fellow of the American Institute for Medical and Biological Engineering in 2021. And also in 2021, he was the recipient of the Mass General Brigham Innovation Discovery Award. So very warm welcome, Jinjun. Just a few quick points before we begin. Please notice that this meeting is being recorded. Please do post your questions in the chat as and when you think of them, and then Jinjun will address them at the end of his talk. And at the end of the talk, there'll also be an opportunity to raise your hands and, ask questions verbally if you like. But now, please join me in welcoming Jinjun for his talk on RNA delivery for cancer immunotherapy and beyond. Jinjun, over to you.
Well, thank you, Dr. Hanking, for so nice introduction. And I just learned that this is the first seminar of this distinguished seminar series. So it's truly an honor to kick off this. And then it's really my pleasure to do this today with you. And I'll be happy to share some of our recent research work with you all today, and with the focus on RNA delivery for cancer and then for atherosclerosis. So let me get started. And so as Anna mentioned, we are in the Center for Nanomedicine at the Brigham Women's Hospital, and certainly Brigham is the affiliated hospital of Harvard Medical School. So the center was established about like 6-7 years ago by Dr. Armindo Faloxar. In the middle here, his laboratory is being, certainly he's also one of my commenter when I did my postdoc training. So his laboratory has done tremendous work with the development and the translation of nanomedicine, particularly targeted nanoparticle delivery for cancer and other diseases. And also he has been focusing a lot on the nanobio interactions to understand how the nanomaterials capture the serum proteins. So he's actually now just affiliated to the center and become a full-time CEO of his own company. And we have another lab with Jeffrey Kopp. His laboratory is focusing a lot on the biomimetic adhesive development, stem cell engineering, and biomedical devices. I also have done a lot of tremendous translational work. And then also my laboratory, it's more focused on big molecule delivery, like biomicromolecules, RNA and protein. And also we are very interested in immunotherapy to combine the RNA technology and together with the immune checkpoint blockade, which I will talk about a bit later on. And certainly we think the backbone of the lab is the materials and then engineering. And so we will combine these different principles together to develop new nanoparticle platforms for better and safer drug delivery. And also the center has some new laboratories now with new PIs, and it's growing fastly. So let me now move to the talk we want to present today. And messenger RNA. So first of all, and I bet over the last 2 years, this is probably the most frequent, if not one of the most frequently used words by the news, by reports, everywhere in the world. And not just scientists know messenger RNA, and a lot of other people now, if not everybody, know what RNA means. So so we know that a very quick background of a messenger RNA. It's in the between of DNA and protein. So we know DNA is our gene information, codes our genes, and protein is plays the function, right? It's the basic functions of to build up the cells and other, important functions in our body. And in between is the messenger RNA is to guide the So first of all, it's guide the translation from the gene information of DNA to the proteins. So it's very critical. And messenger RNA was actually discovered somehow, around 1961 by, I think it is a bunch of different scientists. And very interestingly, no one won Nobel Prize. I think part of the reason, I guess, is because, it's unclear who was the first one. Also, probably there's a bunch of different scientists involved with the discovery of messenger RNA. So anyway, so we all know on the right side you can see, messenger RNA starting from the nucleus, so from by transcribed from DNA to become pre-mRNA, and then become the cleave to mature to become the messenger RNA, and then get transported out of the nucleus, getting to the cytosol, and where in the cytosol the mRNA can interact with the ribosome to generate the protein through the translation process. So as we can see here, compared with DNA, messenger RNA has some unique features. So first of all, in considering as different using messenger RNA as a therapy, you don't have to deliver that messenger RNA into the nucleus, right? So you don't have the nuclear barrier, and because of that, you can avoid host genome integration. You can literally avoid the mutagenesis. Also, the messenger RNA has a very rapid effect. So once you get into the cells, the messenger RNA, you can see the translation of protein expression very rapidly within a few hours. And this translation process is more regulable, and then certainly it's transient protein expression process. So with these unique features, and then because of the the pandemic of COVID-19, and now messenger RNA is well known, and certainly it's now approved, 2 of them for clinical use, including one from BioNTech, and then Pfizer, and then the other one from Moderna here in Boston. So both mRNA vaccine, they use the lipid nanoparticle. So where here is the schematic showing the liposome with the mRNA, though I personally think the structure of the lipid nanoparticle mRNA is much more complicated than the schematic shown here. It may involve a lot of more interaction of the lipid with mRNA, and then form complicated structure within the lipid structure, the liposome structure. So on the bottom is the TM figure I got from Moderna website, and then you can see here is pretty uniform size of the lipid nanoparticle with mRNA. So the question here is, why do we need nanoparticles to for mRNA, right? So let's go back to look at the mRNA intrinsic features, because mRNA is highly negatively charged, so it's literally impossible for messenger RNA to get into the cells because of the charge repulsion. And also messenger RNA is large, so it's like big. Some of them is even a few thousand nucleotides, so it's really large molecule. So again, let's add another challenge for the delivery of a messenger RNA by itself into the cells. And even some of messenger RNA could get into the cells, like for example, by the non-specific endocytosis. Then the enzymes within the endosomes, lysosomes, they can easily degrade the messenger RNA. So not even mention about how the messenger RNA can escape from the endosome into the cytosol, where messenger RNA can be translated into the protein, right? So with all these challenges with messenger RNA, but the promising of the features of messenger RNA. So nanoparticle is now, at least for now, is a perfect match to deliver mRNA. So that's why with nanoparticle we can see you can get a much better cellular uptake, and then also certainly can protect the messenger RNA from the degradation by enzymes. And then also a very important function of lipid nanoparticle or other nanoparticles is to facilitate the endosomal escape of the RNA into the cytosol. and then they can translate it into the protein expression. So the success of the mRNA vaccine for COVID-19 give us a lot of actually confidence to, well, give us a lot of, I would say, additional confidence, but also more excitement to use this technology for other diseases. So my laboratory has been actually working with messenger RNA for cancer delivery about, like, I think 7-8 years ago, when I just started my faculty position here at Bergen. Initially, what we are more interested in is to use the messenger RNA for cancer treatment. So let me talk a bit about the cancer first, because we all know cancer, if not all of them, probably most of them are involved with genetic mutations, and particularly, be involved with the overexpression of oncogene, as well as the loss of function of a tumor suppressor. So when the balance of this oncogene tumor suppressors are broken, then the normal cells will become tumorigenic. So it will, lose the control of the cell growth, and then also will become more metastatic. And then even in many cases, it becomes resistant to treatment. So we think, both are very important. And certainly current pharmaceutical companies, if you look at their pipelines, all those approved drugs, many of them are, focusing on the development of, therapies like small molecules, antibodies, or other strategies to target the overexpressed oncogenes, or more broadly, the pro-tumorigenic drivers. Still in that case, many of them are still considered as, undruggable, and certainly that's an opportunity for silencing technology like siRNA or RNAi technology to target those, oncogenes. But nonetheless, that we see a lot of success with the oncogene targeting. But on the other hand, these tumor suppressors are frequently lost, or lost the function, or complete depleted. In some cases, they are mutated, but at the end of the day, many cases they lose the function of the tumor suppressor, and that we don't see many success yet. Stories yet. So that's how we think, certainly there you can have some ways like to targeting the downstream pathways of some tumor suppressors, or to using the, gene therapy like DNA to restore the, the tumor suppressor function. But over here, as I mentioned about, the unique features of messenger RNA compared with DNA, we do think there are huge opportunities for RNA, messenger RNA to, restore the tumor suppression. So this is how we got started with the use of messenger RNA, and the technology to, work with cancer. And so for delivery of RNAs, we need nanoparticles. Certainly, you can use other strategies, but nanoparticle is certainly, I would say, if not most popular, but I think certainly one of the most popular strategies now for particularly for systemic RNA delivery to solid tumors. And the reason is because we can always come up with a design of different materials, lipids, polymers, etc., inorganic materials. And then through mostly like charge interaction, but certainly you can use all other ways to load the RNA into the nanoparticles, so you can protect the RNA from degradation. And then with the nanoparticles, you can have a better secretion in blood, and with longer secretion in general, you will get a higher accumulation to the leaky vasculature tissues with leaky vascular, such as the tumor tissues. And this is a phenomenon where in the nanomedicine field, we call it enhanced permeability and the retention effect, the EPR effect. The EPR, in simplest way to explain the EPR effect, is because of the, as I mentioned about the leaky endothelial cells, the gap between the endothelial cells, so the nanoparticles can cross the gap passively into the tumor tissues, and then also the lymphatic drainage system is impaired, so the nanoparticles are able to trap in the tumor tissues. But nowadays we all know there are many advanced studies to show like the EPR effect is way more than more complicated than what I just said, and it could be involved a lot of nanoparticle interaction with the stromal cells like endothelial cells. And not necessarily the nanoparticle cross the gap, but they could be potentially cross the endothelial layer through the transcytosis. Also, the nanoparticle, how they interacted with the immune cells within the tumor tissue, and then how the retention over there also has, has some very interesting reports. So, long and short is, EPR effect to me personally, I think is real, though the mechanism is way more complicated than we thought. But anyway, so what we, this is foundation, say for the nanomedicine, to deliver nanoparticles to tumor tissues. And when you get the tumor tissue, certainly you will have, hopefully you can have a better penetration. That certainly is a big challenge in the field. How can we design a nanoparticle to have a deep tumor tissue distribution from the vasculature area? Because we learned that many nanoparticles tend to accumulate around the tumor vasculature, but unable to penetrate through the tumor tissue. But they certainly some studies have shown the capability, depending on the nanoparticle properties, we can have this deep penetration. And then you want to certainly target tumor cells as well as other stromal cells, and then you can come up with a different design either through the surface chemistry or physical chemical property of the nanoparticles to have specific interaction with some cell types. And the other way is you can certainly coat the nanoparticles with a targeting ligand, and then you can take advantage of the ligand and receptor interactions for more specific internalization by the cells you want to target. And then within the tumor tissues, within the tumor cells, particularly endosomes, after nanoparticle getting to the cells, and then the nanoparticles, hopefully, you can use whatever the mechanism we call it, proton sponge effect, or the membrane fusion, and then the nanoparticles can release the from the endosomes to get into the cytosol, and then release the RNA you want to deliver. So this is how it works. It's very complicated, multiple process, particularly for the systemic RNA delivery. So over the years, we have done quite a bit of work. As I mentioned about, we actually initially started with the siRNA delivery, and we come up with a design called lipid polymer hybrid nanoparticle platform. And initially, this is what we designed, and then later on, but the size is pretty big of this nanoparticle. It worked, but in vitro, but the pharmacokinetics of this nanoparticle platform is not as good as we expect. And then we come up with a smaller nanoparticles, which is about 70 to 100 nanometer size range, and then which we call solid lipid polymer hybrid nanoparticles. And then this nanoparticle has much longer circulation, and then much higher tumor accumulation. And then we have been using this nanoparticle for quite a while, and all the stories I'm going to talk today is based on this strategy. And also we have adding additional functions to the nanoparticles, such as targeting ligand, as I mentioned about, to have more specific cell targeting. And also we have incorporated unique functions of polymers, like pH responsive polymers or other stimuli responsive materials into the nanoparticles. So then by doing so, we hopefully can have more control of the endosomal escape and then drug release. Also, we are now developing new generation nanoparticles, and long and short, what we want to do this, is rational design, I call it, is to solve the problem I just mentioned about, particularly for systemic RNA delivery to cancer, to solid tumor. So we want to have a better blood circulation life, and then we certainly prefer to have tissue selectivity. In the case of cancer, then we want to have the more tumor accumulation, and then penetration is very important, as I mentioned. And cell targeting and the endosomal escape, this is all I talk about. And then I think more or less appreciated in the field is how can we control the release of a drug, and how can we achieve more durable activity. So this is something else we are also now working on, and then certainly I haven't published anything yet. So with that, I will now move on to the platform I just mentioned, what they call lipid polymer hybrid nanoparticle platform. So the way we formulated these nanoparticles is actually, I would say, is quite robust. And first, so number one, what we first step, I would say that way, is we just simply mix two different solutions together, which is one is the mRNA solution in water, and then second one is the ionizable lipids. Here this is the one we call the G0C14. We synthesized in the lab, and together with the polymer here we call the polylactic galactic acid, which is certainly a widely used polymer. It's degradable, and we mix them together in an organic solvent, for example, like a dimethylfluorine. And then this organic solvent is miscible with water. So when you add this aqueous solution of mRNA into this DMF solution with G0C14 PLGA, the charge interaction between the ionizable lipids and then the mRNA, they can form quite small complexes within the organic solvent. And then you added these to water solution, and the PLGA is hydrophobic polymer. They will precipitate out to form nanoparticles, but meanwhile, these ionizable lipids, which is amphiphilic, they will also be trapped within these nanoparticle core through the lipid tail interaction with polymer. So it's more hydrophobic interaction. And then meanwhile, on the surface, the lipids or lipid pack will like to self-assemble on the surface of the nanoparticles to stabilize the nanoparticle, more or less like a surfactant. So that's how we can get these nanoparticles robustly. And the size we run, 125 nanometer. And this, I think, this TM imaging shows that. And then also, it's nanoparticles quite stable. And so with this nanoparticle, a few things we'll be able to achieve is to enhance the cellular uptake. Also, and then to improve the endosomal escape, meaning we can see pretty good mRNA-based protein expression, and then in vivo we can prolong this PK, and then because of that, and certainly we can see a higher tumor accumulation of the nanoparticles. So the two tumor suppressor I mentioned about, we were interested in targeting tumor suppressor using mRNA. The two tumor suppressors we have been working on as the two most, I would say, probably most popular ones. The P53 is I would say is most widely mutated tumor suppressor genes in many type of cancers. And then so showing here is a bunch of different cancers and how much they mutated. In certain type of cancers, you can even see 80% mutation. And this is a very good review article by Dr. Scott Rowe from Memorial Sloan Kettering Cancer Center. Certainly, he's an ex-lead pioneer in the field for P53, showing these tumor suppressors must regulate a bunch of different important biological pathways. Like apoptosis, cell cycle, angiogenesis, and metabolism, etc. So literally, I think P53 is involved with many important pathways. And P10, on the other hand, is not as widely mutated as a P53, but also in certain type of cancers is significantly important. For example, in prostate cancer, just to give you one example here, is the homozygous deletion of P10 is very common, around like 40–50% of advanced stage prostate cancer. And so not only these tumor suppressors are involved with the tumor initiation or metabolism, metastasis, also they contribute to drug resistance. So there's a few papers I just listed here is P10, for example, loss of function is highly correlated with the chemotherapy like PI3K alpha inhibitor, and also with the immunotherapy, which I will talk about that later on by the cancer discovery paper. So with that, with the platform ready and with the tumor suppressor we are interested in, we first tested these P10 restoration for prostate cancer, and so you can see here. The green color indicates the expression of a P10 protein, and you can see, with the P10 mRNA nanoparticles, we see that, and then with control nanoparticle, you don't see the green color. The restoration of P10 induced the apoptosis, and then through the biological pathways, traditional pathways, through the PI3K AKT inhibition, and also we can see the dose-dependent cell viability. in both P53, P-link cap cells, the two prostate cancer cell lines with loss of P10. And you can see with the increasing of those, you can see the more toxicity. And but very importantly, I'd like to emphasize here is, when you give this P10-MR nanoparticle to P10 expressing cells like prostate epithelial cells, normal ones like PREC, or to the prostate cancer cells also express P10 like DU145, we can see we don't see these those dependent toxicity, and certainly you can see a better toxicity, but I think this could potentially becomes from either the nanoparticle itself or some transient effect of the P10 overexpression. So that's important because then that's give us the opportunity to specifically targeting P10 lost cancer cells, right? So that's why we emphasize a lot about the precision nanomedicine nowadays, and this we think is a potential strategy to target the P10 specific or tumor suppressor specific pathway pathways. So we then move that to the in vivo studies, and this is a subcutaneous model of the human prostate cancer with P53, and when we give it like multiple injections by intravenous administration, and we can see a good inhibition of the tumor growth, and then also certainly that improves the survival of the this mouse with the prostate cancer. And then on the bottom, you can see here, we can see very good P10 staining, in the tumor tissue. And also we did this testing in a bone metastasis model, which is actually experimental metastasis model. And the reason we want to do that is because bone mets is very common in later stage cancer. Around like 80 to 90% of prostate cancer patients will have bone metastasis later on. So what we saw is here again, we just injected these PC3 cells. By the way, the PC3 cells is where we actually express the cells with the reporter gene called luciferase. And then so you can look at the cell growth by imaging system, by luminescence imaging. So you can see bright color, signals indicating more cells in the injection site. So we're injecting these tumor cells to the tibia, the bone of the mice, and then after 5 days, we started the treatment by intravenous injection. Again, you can see here with control nanoparticles or with control group, you see very rapid growth. With the P10-MR nanoparticles, you can see pretty good inhibition in the bone model. So this is a proof of concept study. And as I mentioned a bit earlier on about the contribution of P10 to the resistance of immunotherapy or the chemotherapy, and that's just when we started working with the P10 mRNA project, we saw the paper published in 2015 by a laboratory in UT Southwestern, Patrick Hu's lab, and his lab did the clinical study using I think it's melanoma patient samples. So those patients actually have received the immunotherapy, particularly immune checkpoint blockade, like anti-PD-1, anti-PD-L1, anti-CTLA-4. So and then suddenly they noticed some interesting phenomenon because immunotherapy doesn't work well for all the patients, right? It certainly works well for subpopulation patients. And what they found is very interestingly on top of these two panels, you can see here, number one, they look at the P10 expression and then correlated to the response of tumor response to the PD-1 therapy, anti-PD-1 therapy. And clearly you can see here with P10 present, you can see better response with the loss of P10, you can see the growth of the tumor in patients. And meanwhile, they also studied the CD8 T cell levels in tumor site, and very clearly you can see here with the presence of P10, the CD8 T cells expression is much higher. So following that work, actually, there are a few other papers published over the last few years, and including 2 papers by Von Allen's lab and Tolani's lab from both from here around the Harvard Medical School, I think it's from Dana-Farber Cancer Center. They studied, I can't remember, this is a very rare cancer, and then this one is breast cancer in a clinical cancer research paper. And in all the cases, what they found is similar phenomenon. Like PTEN loss is associated with the decreased T cell infiltration, which increased the immune suppressive cytokines. And then at the end, you can see very clearly this poor response to immune checkpoint blockade, like PD-1 under PD-1. So that actually, these clinical studies have motivated us to, hey, now we have these PTEN mRNA nanoparticles, we can restore PTEN expression. What if we can combine these two together, and then with immune checkpoint blockade therapy, whether we can see anything interesting? So that's how we now move from human cell line like PC3 to mouse cell line of prostate cancer and melanoma. So now we use a PTEN kappa A, which is is prostate, is mouse prostate cancer cells, and then B16F10, which is the melanoma mouse cell line, and then particularly B16 as a PTEN mutated. It has still PTEN, but it's PTEN loss of function, and PTEN cap cell line is completely loss of the PTEN expression. So in both cases, you can see it's again dose dependent cell toxicity. But then we want to study further. See this cell death has anything related with immunogenicity. So here we will call it immunogenic cell death. So we check the markers of the ICD immunogenic cell death. So for example, calreticulin, which is the if the cells when they die, they can be more immunogenic, and then they like to express this protein on the surface of the cell membrane. And also we look at the release of ATP and then HMGB1. In both cases, indicating that both markers indicate the cell death is associated with immunogenicity. So in both cases, in all three biomarkers, you can see very good expression of the after PTEN mRNA nanoparticle treatment. So so we want to study a bit further, see how these PTEN restoration induce the immunogenic cell death. So there's a very important biological pathway called autophagy, and this is not our original discovery. This actually has a lot of literature have reported already. Autophagy activation is involved with the immunogenic cell death. Also, it can check the anti-tumor immune response. So here we just want to correlate this PTEN restoration together with autophagy. And what we can see here very clearly with the treatment of PTEN mRNA nanoparticles, if you look at the third lane here, very clear PTEN expression. And then you can see very dramatic increase of the LC32, which is the biomarker for autophagy pathway. And very clearly you can see here, by the way, when we add some inhibitor of the autophagy pathways like 3-MA small molecules, you can see the doesn't change the PTEN expression, but does reduce the LC32 expression by these the tumor cells. And then very clearly for ATP is even more dramatic change with the addition of the autophagy pathway inhibitors. You can see much less ATP release. And then similarly, you can see less expression of CRT, calreticulin protein on the membrane surface when we combine the mRNA nanoparticles with 3-MA. So at least from this study, we can see the autophagy pathway activation by PTEN restoration is partially contributable to the immunogenic cell death. And we further tested the cell viability. So for example, if you look at the middle one, which is PTEN mRNA nanoparticles, you see some quite a bit of cell toxicity. But when you combine that with the inhibitor of the autophagy pathway, here this bar here, you can see less cell death. But on the other hand, if you combine PTEN mRNA nanoparticles together with rapamycin, which is the autophagy activator, you can see much better cell death when you combine them together. So this also suggests that the autophagy pathway is involved with the cell viability and then with the immunogenic cell death. And then with those in vitro studies, we move that to in vivo to make sure, this is the case, and, also in mouse models. So we did melanoma models with, and then also the P10 kappa A models in vivo with, here melanoma, I think we did the subcutaneous model. So after only 3 injections, IV injections of the nanoparticles after 3 days, we can see, clearly some inhibition of the tumor growth already after 3 cycle injections. And then we then check the different cell markers like the CD8 positive, and then with interferon gamma positive or the T BET positive CD8 T cells, which both cases indicates the affected T cells, affected cytotoxic T cells. And you can see pretty good improvement of the CD8 T cell, affected CD8 T cell expression of the P10 nanoparticles. On the other hand, we look at the immune suppressive T cells, like the T regulatory cells on the top, and then also these monocytic melanoma-derived suppressor cells. And then we can see again very clearly the downregulation of this regulatory cell expression in the tumor tissues. So indicating P10 as treatment is good to check the immune response in the tumor tissues. We also tested different cytokines, again consistent with what we saw with the cell markers and then the cytokine markers. We can see for pro-inflammatory cytokine, we can see the increased levels. For anti-inflammatory cytokine like TGF beta, interleukin 10, we can see the decreasing of the of those cytokine expression. Then we tested the efficacies. Again, we just used the 3 doses every 3 days. And then we also added a day after the IV injection of the nanoparticles, we do the IP injection of anti-PD-L1 antibody. And then you can see the combination effect from the nanoparticles and anti-PD-1 is much better than just nanoparticle itself, and then even better than the anti-PD-1 itself. So here, this is a melanoma model B16F10. As I mentioned, this model actually is resistant to immunotherapy treatment. So we then just want to confirm the in vivo P10 nanoparticle treatment also induced the autophagy pathway like LC32 with a red color. We can see very clearly the change. And then also we look at HMGB1 in the tumor tissues. We again seeing a very similar trend, like the increasing of the release of HMGB1 from the tumor tissues. So again, suggesting the activation of immunogenic cell death. And then we look at the also the CD8 T cells, and then this is the myeloid derived suppressor cells, which is immune suppressive cells. And in both cases, we can see with mRNA nanoparticle itself or with combination with the anti-PD-1. In both cases, we can see the increasing, and for CD8 T cells, but then for the MDSC cells, we can see the decreasing. So another important thing we want to confirm is this anti-tumor effect is from the CD8 T cell response. So we depleted the CD8 T cells by using anti-CD8 antibodies, and you can see here. So we don't see any T cells anymore, CD8 T cells anymore in mouse. And then we started growing the tumor, and then we give an IV injection of this either P10 nanoparticles with PD-1 without the depletion, or in the red color indicating with the depletion of the CD8 T cells. And then you can see very clearly here with the depletion of CD8 T cells, the tumor growth is much faster now, indicating again T cell response, CD8 T cell response is critical for the for this synergistic effect that we saw in melanoma models. And then consistently, we did this combination in orthotopic prostate cancer models, and then also we did this in as a rechallenging. So after we do the orthotopic model, then the treatment that we selected this completely responsive mice, and then we do the subsequent challenge, and then we can see with the complete response of the those mice, they don't grow the subcutaneous tumor. But for naive mice, you can see the very rapid growth of the subcutaneous tumor from those mice. So again, suggesting, this anti-tumor immune response is systemic and then can actually inhibit the second challenge. So this is a story we mentioned about this. So we come up with the design of the mRNA nanoparticles, something a little bit different from the previous work, but very similar. And then we use the nanoparticles to mRNA nanoparticles to restore the PTEN expression, which can induce the cell death, which is in many cases the immunogenic cell death, and then the release of these biomarkers like ATP, HMGB1, and then calreticulin protein. All these proteins are indeed really called a damage-associated molecular patterns, so they can actually activate the immune response. Also, we haven't really tested these autophagy release, but according to literature, autophagy pathway activation can trigger the autophagy release with antigens, and this antigen can actually activate the antigen-presenting cells like dendritic cells. So then they can activate the T cell response, and with combination with anti-PD-1, that's how we saw a anti-tumor effect. So this is a PTEN story, and then so we're thinking, whether this strategy could be also used for other tumor suppressors in combination with immune checkpoint blockade. So we tested with P53. And by the way, for here, this cancer model, we switched to hepatocellular carcinoma, HCC. And this model, actually, over the last couple of years, we just got a very exciting news. The anti-PD-1, anti-PD-L1 combination with anti-VEGF has, been approved as a first-line treatment for HCC, which is pretty good for the patients indeed. And then anti-PD-1 with anti-CTLA-4 was also approved as a second line for HCC. Very likely, I think, according to the results, it could be potentially also be approved as a first-line therapy for HCC. So, but the question is, again, very similar to other cancer types. There's only some subpopulation of patients, they have this good response to this immune checkpoint blockade therapy in combination with others. So there's still a significant need to activate or to make this immunotherapy better for treatment of HCC. So similarly, we come up with the same design, almost literally saying, we certainly screen the different GCOs, CM, amino lipids we synthesized in the lab. Also, in this case, we put a conjugate ligand called a peptide, small peptide, to target the CXCL4 on the hepatocellular cells. And so according to the, looking at just biodistribution data, you can see here with the targeting ligand, you can see this, by the way, this is orthotypic models of liver cancer. And then we can see, better, not dramatically, but it certainly is very clearly as better than the non-targeted nanoparticles in terms of the mRNA accumulation in the orthotopic tumors. So then we pretty much do similar things. And look at the tumor, give it 3 or 4 injections every 3 days, and then look at the tumor microenvironment change, and then we certainly check in different tumor immune cell markers, and then cytokines, etc. So very clear, interestingly, is some difference you can see here by P53 mRNA nanoparticles itself. It does not induce much change of the immune cells like CD8 T cells, like CD4 T cells, etc. Only when you combine these P53 nanoparticles with anti-PD-1. By the way, anti-PD-1 does not induce much change either. But when you combine these two together, you can see big change in terms of the activation of CD8 T cells, CD4 T cells, and then different cytokines. So this is a very interesting biology phenomenon. We haven't really understand it yet, and we are doing a lot of mechanistic studies, including sequencing, single cell sequencing, and tumor tissue sequencing, and then hopefully we can update these biology mechanism later on. But long and short is so this work is certainly more like technology development and then testing the concept of the combination for HCC. So again, we can see here when you give I think it's 4 injections here every 3 days, we can see very good inhibition of tumor growth certainly they come back after when you stop the treatment, and then they prolong the survival. And also very importantly, actually reduce the metastasis of the HCC to other organs, particularly to the lung here. So that's something when we talk about the mRNA. Now I'm gonna take another 3 or 5 minutes, switch it to a technology called RNA interference, RNAi, and this technology is actually as the opposite of the messenger RNA. Here, we want to use this strategy to silence proteins. You don't want it to have it. So again, this is a very well-known technology. You all we know that, and it was discovered in later 1990 by two well-known scientists, and then later on they won the Nobel Prize in physiology and medicine. By the way, early 2000, the first synthetic siRNA was reported to silence the gene, and then 20 years later, 3 years ago, we have this FDA approval of, now it's 4 years ago indeed, FDA approval of first siRNA lipid nanoparticle called Onpattro for a genetic disease in the liver. As I mentioned, this is the used siRNA, this is lipid nanoparticle also. But then later on now, there's now 3 more new drugs of siRNA approved. And those siRNA actually they use more chemical modification and ligand conjugation, all targeting liver tissue. For I think most of genetic disease and one for metabolic disease. So it's becoming very promising, this strategy. But again, similarly to messenger RNA, siRNA is also big, not as big as mRNA, but it's still much bigger than small molecules, and it's highly negatively charged. So really, cytosolic delivery is still challenging. Enzymatic stability is not a problem now because the pharmaceutical companies, they can do a lot of chemical modification with siRNA, and they can make it very super stable. So, but then the concern is now how we can deliver siRNA to extra hepatic tissues like other heart, tumor, lung, etc. Right? And then how can we improve the cytosolic delivery? So that's what we started collaborating with the laboratory from Columbia University in New York City and Dr. Everett Bass. So he's an expert in atherosclerosis area. And so we all know atherosclerosis is certainly as a big concern for the cardiovascular disease. And so the procedure is, as we all know, the fats start to build on the vessel wall. And then, in small but certain population patients, this plaque, in many patients, the plaque is pretty stable, but in some patients, the plaque keep growth, become bigger and bigger, and then eventually, the cap of the plaque they ruptured, and then they release all these dead cells or the fat into the bloodstream, and then they can induce a thrombosis, and that's how we get a heart attack, we get a stroke, etc. And then even sometimes in some cases, it's deadly, patient, sudden death. So this is certainly a very big concern for the patient. It is certainly always the number one disease, cardiovascular disease. So Dr. Tabas's laboratory actually discovered an interesting biological pathway associated with the macrophage in the atherosclerotic plaque tissue. By the way, macrophage here in the plaque tissue has a very important role in this process called efferocytosis. So what is efferocytosis? So efferocytosis is that macrophage can eat those dead cells, and these apoptotic cells in the plaque tissues. So the macrophage has a function to clear them. So that's how we can stabilize the plaque. But in a certain patient, as I mentioned, the macrophage lost this function. And then there's a genetic mutation called the CAMK2 gamma, which calcium calmodulin dependent protein kinase 2. And in advanced stage of atherosclerotic lesions in human, and then also as well as in genetic mouse models, Dr. Tabas's laboratory found this pathway, CAMK2 gamma, is actually overexpressed. And then that contributed to the loss of function of efferocytosis of the macrophage. So with that, and the macrophage, cannot clean the data cells, and then these data cells build up this plaque, and eventually it will rupture. The plaque will rupture. So unfortunately, there's no very specific small molecule drugs to target the CANK2 gamma, which is a subtype of the CANK2 family. I think they have four different, 3 or 4 different subtypes, and then all the small molecules, they probably pretty much they target all this CANK2 proteins. So that's we talk about the collaboration. This I think RNA strategy has a specificity. We can design a sequence and can only target the CANK2 gamma subtype. So that's how we collaborated together over the last few years, and then we come up with a very similar siRNA nanoparticle, this lipid polymer hybrid platform. In this case, we now coated nanoparticles with the macrophage-specific ligand called stabilin-2 peptide. And by the way, the stabilin-2 is a receptor on the macrophage surface, and then the peptide can specifically recognize this protein and induces cellular uptake. So we come up with a design of nanoparticles, and we tested these non-targeted nanoparticle versus targeted nanoparticles in vivo, and look at the distribution in the plaque tissues. And then we can see here with targeting ligand, you can see about 2-fold increasing of accumulation. And then also certainly you can see a lot of accumulation to the macrophage cells, but not these other cell types. So with that, we then tested the CANK2 gamma silencing, which certainly shows in both cases, the mRNA level or protein level, we do see pretty good silencing. Also, we look at the another very important marker, so we call the MERTK, which actually is a receptor very critical for the efferocytosis function. We can see with the downregulation of CANK2 gamma, we can see the upregulation of this MERTK protein, indicating you will expect to see better efferocytosis. And then this is how we saw this efficacy data after 4 weeks continuous injection every 3 days. We can see that number one, the core area of this plaque tissue area here, you can see this the white one within these blue markers. You can see smaller size in the cross area, and certainly you can see also the cap thickness increases, which means they can protect the stability of the plaque better compared with the control groups. So I think this is all I'd like to share with you today. So we have developed a bunch of nanoparticles for systemic mRNA or siRNA delivery to solid tumor, and we have used these nanoparticles to restore tumor suppressor such as P10, P53. We found the combination of P10 or P53 with anti-PD-1 can have better immune response, can have reprogram the immune microenvironment for better cancer treatment. Also, we have using these siRNA nanoparticles to target the macrophage to silence the CANK2 gamma in macro, in the atherosclerotic plaque tissue, and that can actually promote the plaque stability. But again, overall, we are more like platform laboratories, and we collaborate with biologists and with physicians, and this is actually all the collaborative work between different groups. And then indeed, with Bruce Zeta and Dan Duda, with Chuck Bieber-Rich, we do all this cancer work. And with Dr. Hong from Children's Hospital, and then Dr. as I mentioned, Eric Tabase, we did the immunotherapy work. And also the team, which they did all the work, not me. And Arif did the P10 work, and Yaoxin and Yi, they did I think they did the combination of P10 with the immune checkpoint blockade. Yulin did the combination of P53 with the anti-PD-1, and Wei did the siRNA work for atherosclerosis. And I also like to acknowledge the funding support from different institutes. And I think at the end, I would like to close my talk by again borrowing one slide from Moderna. We really think this is the beginning of RNA therapy. Just think about the potential what they can do. Using siRNA, you can literally silence almost any gene you want with messenger RNA or other type of RNAs, you can translate to, different proteins, intracellular protein, membrane protein, secreted proteins. So it's huge potential, and this is the area. It's just the beginning. Well, thank you very much. I'm happy to answer your question, and I apologize for the for the long talk.
Not at all. Thank you very much.
Thank you,
Jinjun. On behalf of my whole department, thank you. And look, there's lots of virtual applause coming for you from our audience. This is this is very far from my own research area, so this is completely fascinating for me to listen to. And you are having lots of questions flood in the chat as we speak, actually.
Okay. Anna,
surprisingly from Rongjun.
Anna, can I just say, Jinjun, can you hear me?
Yes.
Yeah, I just wanted to say many thanks as well. That was a great presentation. I actually have to go in a minute to another meeting, but I just wanted to say thanks a lot for joining us and for giving us such a great talk. And I hope we can do some work together in the near future. I think it would be great.
Okay. Well, thank you very much, and I'm certainly looking forward to meeting you in person too.
Yeah, absolutely.
Okay. Thanks.
All the best. Cheers.
Yeah.
Bye.
Hey, Rongjun, I could read all of your stuff out, but I invite you to come to appear online to ask your questions yourself, if that's all right.
Okay, thank you, Anna. And thank you very much, Jinjun, for your very interesting and very inspiring talk. I really enjoyed it. And first of all, I like the system, your solid polymer lipid hybrid nanoparticle. This is very interesting. I think it's very good to have solid core, a lot of advantages. I have one question here. So, you use PLGA. This polymer is relatively hydrophobic.
Yes.
Do you think if the coexistence of PLGA with lipid RNA complexes in the nanoparticle core, would that compromise the RNA encapsulation efficiency and also the RNA release capacity?
Oh, that's a very good question, Indeed, we deliberately, one purpose we want to, incorporate these PLGA into the nanoparticles. We know lipid nanoparticle work pretty good, right? And then, so the purpose when we started that platform, as I mentioned, that we want to use that for siRNA delivery initially. And one thing we'll think, when you incorporate the polymer, you probably potentially can prolong the release of siRNA from the nanoparticle. So that's actually that's exactly what we saw, and then we published a paper in Nanomedicine after that, and that shows that we can prolong the silencing from in general like weeks to 2 weeks. So that's something what we think is a benefit, and then another benefit is certainly we had a thought we can certainly also use this strategy to load the hydrophobic drugs. So we can do combinatory delivery of RNA and then drugs together. So we indeed we haven't published anything that although we had that idea, but I agree with you with the polymer. It certainly made the platform more complicated, and number one is make the formulation a bit complicated compared with just lipid nanoparticles, and second. Regarding the encapsulation efficiency, it's possible it's a bit lower than lipid nano, but overall the encapsulation is still pretty good. I think it's 50 to 80% depending on the formulation you use. So overall, I think again the loading of siRNA or mRNA is still depends on the charge interaction between the ionizable lipids and then the RNAs. So PLGA is more or less, more as a core to carry this the complex, and then the interaction, as I mentioned, is through the lipid tails of these ionizable lipids with the polymer PLGA. So we don't see a significant reduction of the encapsulation efficiency, but we do see a bit, but it's not a big concern for us now in that case, and so. But now I will have to say, we are also developing different new version of lipid nanoparticles, and I think there's something we certainly could do better with lipid nanoparticle, and then hopefully I can share more with you later on about our new work.
Thank you very much, Jingjing. I can see a lot of advantages to have some solid core, and I believe that mechanical strengths would play a role in the 3D
Yes.
in the in vivo delivery.
Yes.
My second question is related to what you just answered. So could you please comment on the differences in challenges of delivering small siRNA compared to large mRNA using your solid polymer lipid hybrid nanoparticles?
Yeah, yes. That's I would say, it's a big difference indeed. Though you can see we use a pretty much similar platform, right? But indeed, when we switched it from siRNA to mRNA, we did a lot of optimization. And even we did that, I can tell you back to what I mentioned about for siRNA, we did see the release, peroneal release from the nanoparticles compared with just lipid nanoparticle. But for mRNA, we actually do not see that. We don't see big change, in terms of the durability of the activity of RNA. And so we then thought more about that. We think it makes sense because mRNA is so much bigger than siRNA. So in one nanoparticles, you probably can load like tens or even hundreds of siRNA molecules. But for mRNA, you probably can load a few only to the nanoparticles. So that has given us the big difference. So only a few mRNA, so it doesn't really matter, the release kinetics, right? So more. That's the reason we don't see much of the change in terms of the durability of the mRNA. So again, that's big difference to me, the size, and then now we are switch, as I just mentioned about back to we want to study more actually now the lipid nanoparticle formulation to solve the durability issues. So hopefully we can have a better version of lipid nanoparticle to control, not necessarily through the release, but more to stabilize the mRNA to have a longer activity.
Thank you very much, Jingjing. I have another question, but I think I should stop now because we have quite a few other questions, and we have one-to-one meeting later. So if we do not have the question to our meeting.
Yeah.
Well, let's see how we do for time, Rongjin. We might get back to your question. Right, Jingjin, we have a question from Simon in the chat who says, thank you very much. Does the required dose of mRNA and the toxicity differ from current RNA vaccines? Otherwise, how did you optimize or screen the nanoparticle composition for efficient mRNA delivery? Can you see the question in the chat yourself?
Again, I'm not very good at this Microsoft team. I don't. Okay, yeah, I can see the chat. But anyway, I hear your question. I think it is a very good question. And then the success, as I mentioned about using all the you also use lipid nanoparticle for siRNA, messenger RNA. So lipid nanoparticle is very popular. You can tell a lot of publications, a lot of interest from industry, and so in terms of our strategy, and then you can tell for lipid nanoparticle for siRNA, the design of those what we call the LNP platform, more or less for liver targeting, most of them. And then for the vaccine side, you certainly avoid this systemic IV injection, systemic delivery, so it's more local injection. And I don't know, whether you switch a lipid nanoparticle, certainly for the Alnylam's, Onpattro design of lipid nanoparticle with the this lipid nanoparticle mRNA vaccine, they have some difference. First, number one, I think these ionizable lipids is different, and the other components are pretty much similar to me. So the ionizable lipids certainly, is a better version, and then in terms of translation, I think they work pretty similar. We compared it in the lab, and then certainly those new ionizable lipids in BioNTech or Moderna's vaccine nanoparticle, they have a more rapid degradability. Excuse me, and compared with MC3 ionizable lipids Alnylam used. So I don't know if this platform could be used for cancer because if you can see, you do the IV injection, they have pretty much very rapid dissociation of the lipid packet from the lipid nanoparticle. and then they can absorb a lot of like interesting serum protein, like ApoB, I think, if I remember correctly. And then this ApoLipo E, I think. ApoLipo-B or ApoLipo-E, I can't remember. But then those serum protein then can target the hepatocyte cells. So these platforms are very good for liver targeting. And then for cancer, we know that you need a little bit better circulation. So DMG, by the way, they use all these either DMG or similar lipid pack, which have 14 carbons linked to PEG pack. Those, as I mentioned, this lipid pack can easily dissociate from the LNP, and that is certainly won't give you very long circulation in the blood. So that's why we think we need a new version of nanoparticles to target the cancer. And whether we need the hybrid platform, or we can just modify the lipid nanoparticles using different lipid pack, different components, that's a different story. So but long and short, I think with current FDA or clinically approved the lipid nanoparticles, they may not work. We may not be. They still need the optimization for cancer targeting or even for other diseases.
Okay, thank you very much. And there was a follow-up point in that same question of have you characterized the surface charge of your nanoparticles to see if you could tweak it to affect the biodistribution?
Yes, absolutely. We have done this. We actually using not for mRNA but more for siRNA. We screened a bunch of different lipid pack. As I just mentioned about with longer tail like DSP pack, you have a longer circulation. With shorter tails, you have a shorter circulation. And reason is, as I mentioned, this lipid pack can dissociate from the nanoparticle surface. And longer tail, you have more hydrophobic interaction, so it's slower. Shorter tails is much faster, so that actually rapidly changed the surface charge from initially, around neutral charge or slightly negative charge to positive charge. So that's why what we saw is very interesting. We published a paper in Theranostic many years ago. What we saw is, with longer circulation in nanoparticle, you can have certainly better tumor accumulation and then silencing. But again, this is you need a balance, because with the more stable pack, you have less cellular uptake indeed. So it's really a balance you need to find, because doesn't necessarily mean, very stable lipid pack on the surface will give you the best outcome. I do think, because you have one hand, you have a long PK, high accumulation, but only hand you have less uptake, and that's going to be useless. So it's really a very quick question. I think, we have done a lot of, again, this is just, you have to do a lot of screening, I think, to find out the underlying, the balance.
Okay, thank you very much. Right, so our next question is from Alessio, who is in Omar Marta's group. So his question is about flow. He says, thank you very much for the very interesting talk. Just a question. Have you tried or do you plan to use continuous flow mixers, such as confined impinging jet mixers, for the nanoprecipitation process?
Oh, this is a great question indeed. I love the question a lot because, my previous mentor, by the way, I have to say that when I did my PhD training, I did a lot of microfluidics. So, and then when I came here as a postdoc training, I sort of like give it up. But I do think, and there's a lot of papers already published indeed in the field. Microfluidics or other strategies like, engineering strategy, the flow-based mixing, they are very good indeed. And then if you look at lipid nanoparticle, that's a standard. Use either microfluidics or other mixing flow-based mixing strategies to synthesize nanoparticles. So in all case, I think we have not done that, but I think based on what I learned, it could give us some problems, because when we work with a PLGA polymer, somehow, particularly for the microfluidic channels, the PLGA will tend to somehow precipitate it out after many cycles synthesized, and then they're going to block the channels. Lipid nanoparticle doesn't have this problem. And then I think you mentioned about the jet mixer. I think that could be a very good strategy to solve the problem indeed. Again, but we haven't done that. But I think that will give us better nanoparticles, more uniform, smaller size. So it's a great question. Thank you.
Okay, thank you very much. Then next there is an expression of admiration for you from Noor, who says, Professor Jinjin, I can't express how thankful I am for this interesting and fascinating presentation. I enjoyed every second of it. In fact, I have some ideas in this sense that can be computationally aided for my PhD project, and I want to know if I can contact you for some expert guidance and perhaps collaboration. Thank you so much.
Oh, you're more than welcome to do that.
Okay, thank you very much for that. Can I
you.
ask a question now on a completely different level? So you showed on several slides graphs which showed the tumor volume growth with time.
Okay.
And on for example, on slide 14, it started almost at 0. And did I understand correctly that was in human tumors?
Give me 1 second. You're talking about 14, right?
14, yes.
Okay, that's a good question. Yeah. By the way, this is yes. Please go.
I mean, my question was going to be, so in all of these kind of results graphs that you showed, the tumors, they all started at the same size. And I just wonder how, in terms of how you do experiments, how do you ensure that it's the same type of tumor? And how do you track it to make sure that you start the measurements from the same point in each sample?
Yes,
right.
Yes,
okay.
Absolutely. It's a good question indeed also. And I've been, many times questioning my my students how, so first of all, I like to clarify that's one thing. We also usually we start at a bit smaller tumor, like 50 to 80 millimeter cubic. So you can see here right here, it's not starting from 0 indeed. It's just because of skill bar, and then you don't see much. It's a small tumor, and then in other cases, for example, here we actually normalize the tumor growth to the day 0, or in this case day 5. So for each mouse, we normalize the signal change from the background here on day 5. So that's why we started from 1 over there. So that's you see why you see exactly same, but indeed it's not. If you can look at this figure, I apologize because there's too many mice here. You don't see it very clearly, but you can see it's actually a lot of variability indeed. It's not like perfectly from same size. So definitely a lot of size change. And then also, I'd like to point it out here, we have not done big tumor yet, which is very important for future study. We all study from small tumor. We know that small tumor may work better in terms of experimental design. But if in the case, for example, if we can grow the tumor bigger, much bigger, like reach it to 500 cubic centimeter, whether these nanoparticles work and how they work, we haven't able to done that yet. So, but I do think that's going to be an interesting question for us to answer later on.
How big a tumor can the mice take before,
certainly you can let it grow for very big, but then we are the policy from the ICUC animal facility, they do not allow the tumor to grow too big because that may be painful for mouse.
Mm-hmm.
And so usually, the limit is 2 centimeter in one largest dimension. Yeah, it's too big. So yeah, but that's gonna be the terminal point. So usually we go to like about 1 to 1.5 centimeter, and then we stop it. So but again, that's good things to keep in mind. If you look at the literature, I think a majority of them they just use small tumor, but then a large tumor like SSM 500 millimeter cubic, that will be probably more meaningful indeed. But again, it depends really how do you think about this, because if this tumor is really large, then in general, in reality, is in any possible ways you're gonna do the surgery first, right? And then you give the drug later on. So that's why we have an argument why we don't want to need the big tumor. But in some cases, you probably are not able to do the surgery, and then that's in those cases, it's very important to test the efficacy in large tumor setting.
Yeah, I understand. And do you always succeed in getting the tumor to form in the same part, in the same organ in the animal?
So I think for subcutaneous model it's easier because literally you just pull the skin and then do the subcutaneous injection. For orthotopic models, it's more challenging and the success rate is lower. and then the tumor growth, you probably will see more variability. Let me see, I can show you one data here. This is the one. This is also topical model, but yeah, it's the beginning. They pretty good. I think this one is. Again, this is probably I'm not sure we normalize to. I don't think we did normalization, but we did start from small ones. So those are I think that's just good. I guess it all depends on the skills of the person who did the experiment. So yeah, it's from here. It looks like pretty good to me.
Cool. Okay, I'm learning some of this. Is fascinating.
Oh, thank you.
Right. Thank you so much. Right, I think we're past your scheduled time with Rongjun. So I think yes, let's just double check. Yeah, I think that's it for questions in the chat. So please join me again for thanking Jinjin for the wonderful talk. It was really, really interesting. Thank you so much for joining us.
Thank you. I really, enjoyed talking with you all, and then I appreciate this opportunity. I wish you all, everything going well with everything you're doing. So
and you all the best for the new nanoparticle developments.
Okay, thank you.
Thank you very much.
Okay. All right, bye-bye.
Thank you, Jinjin.
I will see you again later.
Thank you for the questions, everyone.
Bye.
Jinjin, you want to take a few minutes break? Oh.